Uh, Here we go. So everybody that's on this call knows me. I will not go through the the effort of introducing myself. Um, but I do want to say a big thank you for being here. It was everybody that was invited to join this conversation and get to hear live the results of some of the research that RPLA has been honored to fund. It's because you have given time and or dollars, significant time and or dollars to the organization to help advance our mission. So this is one way of saying thank you very, very much and allowing you to see what your efforts have allowed us to contribute to. Um, Research and supporting research and helping advance research has always been really central to our mission and to what our vision for Recurrent Pregnancy Loss Association is and can be. The reality is that still 50% of cases of RPL are medically unexplained. And we really are committed to moving the needle on that and giving more families more answers. Um, in addition to our research grant, which 2025 is the fourth year that we will have distributed the grant. There's been just some other big things that I wanted to mention. So hopefully you've been following along in our progress in our monthly newsletters. If you're not getting those, please feel free to email me and I'll make sure you get them. Um, but a couple other big things that I just wanted to make you all aware of in case you didn't know. We this year expanded our support groups. So now we're offering 3 support groups per month. They're all free, they're all virtual. Um, but we've had good attendance and there's increasing demand from the community for wanting those that type of support, peer support. We had our first awareness run earlier this year, late August, early September. Some of you participated in that, but we had about 100 people from across the US and some in Canada that ran with us to help raise awareness about recurrent pregnancy loss and its impact on individuals and families. Um, and this summer, Dr. Ron Labrizzi, one of our board members, and I got to speak at the IPPE Summit. That's the International Partnership for Perinatal Excellence. So that was really exciting. And actually, Dr. Workalemahu also spoke there. So I got a sneak peek of this presentation. And then coming up in November, thanks in large part to Dr. Labrizzi's connections and efforts, we were able to help plan a day-long summit, or actually continuing medical education session with ACOG, which is the American College for Osteopathic Obstetricians and Gynecologists. So there will be a day-long virtual training for clinicians specifically focused on recurrent pregnancy loss, and that's coming up in November. So a lot of things that we're really excited about, in addition to our ability to directly funding some really cool research. A shout out to, because I see their names, a shout out to some of our current and past board members who are on the call. So just to let you know, so Joey Conifal's on the call. He's our current board president. Ben Burnham's on the call. He is the co-founder with me of Recurrent Pregnancy Loss Association. Ron Labrizzi, one of our board members. Michaela Stevenson, another board member. Amy Henderson Riley, a former board member. Oh, Melissa, there's Melissa. Melissa's a current board member. So thank you all for being here and for giving your time and helping so much with this cause. But I will stop talking now and kick off the main event. I am thrilled to introduce you all to Dr. Tsega Workalemahu. So I will give a quick introduction and then let him do the more exciting part of the talking. So Dr. Workalemahu is an assistant professor of obstetrics and gynecology in the Division of Maternal Fetal Medicine at the University of Utah School of Medicine. He has a background in genetic epidemiology and leads a research investigation on genetic mechanisms related to obstetric complications and their short and long-term adverse outcomes. He will present his team's recent findings from population and family-based studies in Utah that investigate genetic factors contributing to pregnancy loss. So I'm really thrilled to be here tonight and listen to this session myself. And I will pass it over to you, Dr. Workalemahu.
Thank you so much. Can you hear me fine? Okay, I'm gonna share my screen. Great, thank you for having me. It's truly a privilege to share this journey with you, a journey that would not be possible without the generosity and courage in this room, as Megan mentioned, and I'm honored to contribute to this conversation and understanding recurrent pregnancy loss. So in this talk, I will start mostly covering on what we have done to understand stillbirth, which we define as pregnancy loss at 20 weeks gestation and beyond in the US. I'll also touch upon recurrent pregnancy loss and recurrent stillbirth, but our project is motivated by not only the alarming burden of stillbirth, which we have better characterized than other pregnancy losses, but also the need to understand unexplained stillbirth, which make up about a quarter to half of stillbirth, for pregnancy loss, as Megan mentioned earlier. So we are motivated by the need to provide answers to suffering families and support bereavement. We also know that many more families face early pregnancy losses, and for many, even after testing, the cause of this loss remains unknown. And that can be one of the most painful experiences. And this work is personal for so many of us. And stillbirth and recurrent pregnancy loss are heartbreakingly common and yet still so poorly understood. And so that's why we're doing this kind of work. Now, there are many risk factors, and most stillbirths have underlying placental causes. But these risk factors pose a lot of challenge for precise diagnoses in the clinical setting, and they are often identified late when prevention of death is no longer possible. So we know the placenta fuels pregnancy, but when it doesn't develop properly, it can lead to pregnancy loss. And we know the placenta is critical for nutrition and oxygen transport to the baby, but it's also increasingly being recognized for other mechanisms like cardiovascular remodeling during early development and having a lasting implications on the health of the baby and the mother over their lifetime. But what's fascinating is that most of the placenta carries the baby's DNA and not the mother's. So inherited genetic differences from both parents can influence how well the placenta works. But many of the prior studies of pregnancy loss have ignored paternal DNA. And when we did a systematic review of these studies, mostly what we've noticed is there's very little effort to include the mother, the father, and the offspring in these genomic investigations of pregnancy loss. Among those studies included in this review are these studies that are from a well-established case control study of stillbirth that showed genomic technology advances our ability to understand genetic causes of stillbirth. For example, karyotyping diagnosed genetic abnormalities in about 6% of unexplained stillbirths. And this is a technology that's been around for more than several decades. And it helps you look at the chromosomal bands and identify these huge changes in the chromosomes. But when microarray came around, this is a newer technology, it increased the diagnostic yield by additional 8.3%. And this technology allows you to identify deletions and duplications in the chromosomes that you will not be able to identify with previous technology karyotype. When whole exome sequencing technology became available, it identified causes in another 10% of stillbirth, identifying Mendelian disorders, conditions that may be caused by single gene mutations leading to stillbirth. So this clearly shows that more sophisticated sequencing technology reveals previously unidentified causes of stillbirth. But these studies only use DNA from placental or fetal samples, lacking DNA from both parents or broader familial context, missing key contributors like inherited or newly occurring mutations. So I guess the natural question to ask to justify genetic investigation is to ask this question: does stillbirth run in families? To explore inherited risk, we conducted a population-based study using the Utah population database by linking stillbirth records with matched live birth records to 2.5 million family members. And using this data, what we found is that we identified multi-generational families, what we call pedigrees, that had significantly more stillbirth than expected by chance. And these high-risk families may have heightened inherited risk compared to low-risk or unaffected families. So there's increased stillbirth risk based on family history. More specifically, male family members shown in blue had higher stillbirth risk if their first, second, and third degree relatives experienced stillbirth compared with male family members without a family history. And so this is another point that suggests that a paternally linked inherited component that's been under recognized for stillbirth. So stillbirth clusters in families, and there appears to be a paternally inherited risk for stillbirth. And so it's not always clear when you look at maternal risk factors when understanding stillbirth. And given what we know about the importance of placental health, potential risks coming from the dads should also be investigated. So our next step was to identify what those inherited genes might be, and can we find genes in these high-risk families that may be shared between affected individuals, leveraging this multi-generational pedigree data from Utah. So thanks to all of you again for your support through RPLA's support. We were able to tap into one of the kind genealogical database from Utah and sequence just a handful of placentas from stillbirth among these high-risk multi-generational families. And we applied a method known as shared genomic segments analysis. And shared genomic segments analysis allows you to find or search DNA regions inherited in stillbirth, leveraging this multi-generational data. We were fortunate to have our team members here who developed this approach, which aims to identify what's common or shared among these stillbirth cases in terms of their chromosomal regions. So if these sampled cases are sufficiently separated, so distantly separated like you see here, you expect that they wouldn't be sharing disease genes by chance. So this kind of analysis helps us find out whether these distantly related cases share segments of the genome longer than expected by chance alone, pointing us to inherited DNA regions. So using this method, we found shared genomic segments between stillbirth in 3 families in pedigree A, pedigree B, pedigree C, shown here. And when you look at these shared regions, they include genes linked to fetal growth and placental abnormalities. For example, stillbirth in 2 families, pedigree A and B, shared a gene called IGF-1R. Disruptions in this gene are associated with fetal growth restriction, meaning that when the baby is not properly growing, there's a chance that this might be a reason why there's third trimester loss in this case. And stillbirth, another family shared a region that included a family of genes called HLA gene, human leukocyte antigen gene, that helps the immune system tolerate the baby. And if this gene doesn't work well, there's research that shows that mother's body may reject the pregnancy. Also, other stillbirth and other family shared a gene called DLK1 and RTL1, which are paternal origin, and disruptions in these genes affect fetal growth and placental function. Sometimes a condition called Temple syndrome are associated with these genes, which is often undiagnosed and likely fatal in utero. So what does this all mean? This sharing suggests inherited causal mutations may exist in these regions, but we need to carry out targeted validation to confirm and find causal variants. But for us, this is a proof of concept, meaning that without having to sequence the parents in a lot of individuals, this method allowed us to leverage that large multi-generational family structure that I showed and DNA just from a few affected individuals to understand inherited contributions. But deep family structure data is not always available, and other approaches like parent-offspring genome sequencing is required to understand inherited mutations in most cases. Also, not all stillbirths are associated with inherited risk factors. There are other genetic factors that are known as de novo or spontaneously occurring mutations in the offspring that can be contributing to a stillbirth. But we have very limited knowledge of that, and investigating both inherited and de novo mutations in any case requires sequencing the parents and the offspring in most cases. So we were fortunate to have a few family members to participate in our study. We enrolled couples with recurrent pregnancy loss, sequenced both parents and the placenta from the losses and the saliva from the live-born siblings. These are reproductive age couple who suffered repeated losses, including unexplained early miscarriages, stillbirth, and very much wanted to know what happened. And so we looked for genetic differences or variants between babies who lived and those who didn't within and across the families. And we had our graduate student help us with the analysis in our department chair from human genetics, who actually developed the method to help us understand what's inherited and what's false positive and what's true positive and so on. What we found in stillbirth was a newly occurring de novo mutation in a gene called filament B, a maternally inherited a mutation in a gene called myosin isoform C, and a structural variant in a gene called fibrillin 2. And mutations in these genes are all associated with phenotypes related to skeletal abnormalities, which we know are lethal in the third trimester. So it's possible that de novo and inherited variants in the placenta or the fetus may be lethal, acting through skeletal development. But they're not all in the same family. But why we think these might be relevant to stillbirth is that we didn't find these mutations in the live birth or in across any of the families or population controls from public databases. All in all, these two studies that I mentioned together provided a proof of concept informing our next study. And I'm happy to say that these RPLA funded studies helped us secure a major NIH grant to expand this research. I want to say that this is an extraordinary example of how private support jumpstarts transformative work. So our project is entitled Familial and Genetic Determinants of Stillbirth, with the emphasis of families indicated here, with inclusion of both parents and the children. In this project, we have two project goals. We hypothesized that inherited variants may underlie familial or recurrent stillbirth, and the newly occurring mutations may drive these sporadic or isolated stillbirth cases. And we've been enrolling families, and I'd love for you to reach out to one of our study coordinators, Sarah or Danae here, if you'd like to find out eligibility and enroll in our study. And I want to say that we would like to have more patient participation, and we would like to generate quality data to understand causes and catch up to others who have made discoveries in other diseases, and extend that to pregnancy loss, which we know is very much overlooked and understudied. So what's next? First, we want to identify causes to facilitate closure and bereavement and better counsel families based on genetic findings. But also imagine being able to identify families who need early monitoring or using couples genotype to guide treatment, or knowing which pregnancies need intervention before even a problem arises. That's where we want to go. And we want to improve risk prediction and recurrence counseling, but also guide targeted monitoring in high-risk pregnancies. If we know which families are at high risk based on genetic testing, we could provide targeted monitoring. And using the results, we'll also guide the development of new therapeutic strategies. So stillbirth has a genetic basis. Both inherited and newly occurring mutations may contribute to stillbirth. Placental genetics is critical to understanding causes of pregnancy loss. And paternal contributions and imprinting effects are underexplored and may be key to unlocking unknown causes in stillbirth and pregnancy loss. And we also know that emerging genomic technology and family-based studies are shifting that paradigm. So genetic research in stillbirth is not just about science, it's about restoring agency and answers to families. And I just want to say that we have to look beyond just maternal risk factors to find causes. So I'd like to thank my team. We're very multidisciplinary team of scientists, care providers, and advocates for stillbirth and pregnancy loss. And I would like to thank RPLA and our other funding partners, including families participating in our studies. And thank you for being part of this journey. We're just getting started, and because of you, we're getting closer to a future where loss doesn't have to be unexplained, and hopefully a grief can be met with knowledge and compassion and hope. And I would like to leave this contact information if anybody would like to reach out and participate in our study. Thank you very much.
Thank you very much. I, like you said, I think this is exactly what the hope was, right? Is to help provide dollars that can get to that point of proof of concept and then allow for bigger funds to be easier to access. So I'm so glad that it worked that way. We have quite a bit of time left, and so I want to invite anybody that's on the call to ask questions. I know there are a handful of doctors on the call, so please feel free to ask technical questions if you've got them, but a number of people that are personally invested and been impacted by this. So please, any questions that you have about this work, if you do have a question, and if it's possible for you to turn on your camera and unmute yourself to ask it, that would be great. If that's not possible, feel free to type your question into the chat and I can ask it for you. If there aren't any questions, then I'll have to come up with some, and I'm not always the best at it. So putting you all on the spot to ask some good questions.
I have a question. Sorry, I can't turn on my camera at this moment. I was just wondering if you guys had looked at whether or not there were differences based on if people had explained recurrent pregnancy loss or unexplained recurrent pregnancy loss, and you were looking at the differences of their placental tissue and how you might do that in the future when you're designing future studies.
So if I understand the question, using placental tissue for unexplained recurrent pregnancy loss overlaid with genetics, is that the question?
Or if you looked at whether or not they had like a diagnostic evaluation to see if they had an underlying etiology of their pregnancy loss,
yeah,
and how that impacted their gene testing.
Yes, I really like the question. So the criteria actually for our ongoing prospective studies is unexplained etiologies. So pregnancy losses that had a clinical workup, including pathological exams, and in most cases, they've been ruled out in terms of the other risk factors or contributing factors, and a lot of these families have no diagnostic findings for their losses, and so they get enrolled in the genetic testing to determine that. In some cases, they have had some genetic testing like karyotype or microarray testing, and that's been ruled out as well.
Thank you.
I have a question, Doctor, and I apologize if there's background noise, but I appreciate the work you're doing. It's fascinating and wonderful that you're kind of addressing the unexplained is unacceptable part. Do you have any idea how transferable the information you're getting now is to pre-20-week pregnancies, early pregnancy loss? Early recurring pregnancy loss?
We consider losses across the developmental spectrum. There is some, the etiologies vary across gestation. We know that over 70% of early pregnancy losses are explained by these big chromosomal abnormalities and changes in the chromosome. And a lot of that also has dysmorphic chorionic villi that after assessment of the placenta, you see a lot of the early losses which have dysmorphic chorionic villi issues. So between stillbirth and early losses, we're still far away from identifying shared etiologies. I mean, that's going to be our next step to try to understand embryonic lethality and things like that that might be related. But the etiology is just so different across gestations. But we're exploring across the developmental spectrum to understand the shared genetic etiologies at the moment. We just need more data.
Amy, go ahead.
Great. Thank you so much. Super interesting presentation. I'm a social scientist and I work in public health. So your presentation had a lot of words with a lot of syllables that I don't know. But I thank you for that because I've learned a ton from this presentation. So I really have a process question for you, Professor, and that is, where else are the sources of funding for this work? So as a former board member and a donor to RPLA, I'm really interested in more of this work and helping to produce more of this science and more of these answers. So I guess my question is two-part. Where else is there funding for investigations such as this? And how else can we as donors and as part of the RPLA community help you and other scientists who are interested in this work? Thanks so much.
Yeah, we were very, very fortunate to have RPLA jumpstart these pilot, what we call pilot studies. These are small studies. So genetic studies require they're expensive. There's recruitment and there's a DNA sampling and sequencing, the sequencing and the analysis, and the technology costs quite a bit. And the need for a lot of individuals to participate in these studies because we're just not being able to scratch the surface with just very few individuals to understand causes. So there are other funding mechanisms that we were able to tap thanks to RPLA, and we were funded by the NIH to scale up our investigation and expand our recruitment and sequencing. We've had other funding, pilot funding from Star Legacy Foundation for stillbirth and internal Center for Genomic Medicine at the University of Utah that we were able to fortunate enough to do some of this work. So we do want to continue on this, and it really requires partnering with many individuals and having more samples in our data to understand causes.
Ben, I see your hand raised. I'm going to come to you in just a second, but I want to take one step further on Amy's question. Dr. Workalemahu, so as you know, more funding and then more samples. So he didn't talk about it at all, but University of Utah is a site of one of the there's a pregnancy after loss clinic there. And there's a lot of work being done right now in the US to expand and grow a network of pregnancy after loss clinics, specifically where couples can go to receive pregnancy care if they've experienced prior losses, any type of losses. But their clinic specifically sees a lot of infant loss, stillbirth, and recurrent pregnancy loss as well. My question is related to recruitment. Is that a plan with the Pregnancy After Loss Network to use those other locations as a way to get more people enrolled in these types of studies?
Yes, I think you're right. I mean, we have actually recently we became a center of excellence in stillbirth prevention, Utah Center of Excellence in stillbirth prevention, and that effort includes the exactly what you mentioned, the pregnancy after loss clinic, by the way, which includes the services including bereavement support and mental health counseling. And this is very much integrated, and it's not, our project is not all about genetics. It's also to understand counseling based on the genetic findings and what that implicates and how we handle testing results explaining to the families. So it's very much integrated with our study, and we do recruit families from those clinics as well. And we're actually this week, starting tomorrow, we're hosting a workshop on bereavement support, placental assessments, and autopsy. And it's called the Improve Stillbirth Workshop.
Ben, go ahead.
Hey, thanks very much, Dr. Workalemahu. Thanks for your work and thanks for being here with us tonight. I was wondering, would love to hear a little bit more about your kind of what's next. You mentioned that, you know, through this sort of proof of concept, you've been able to secure additional funding. And I wanted to hear about sort of what the objectives of those studies are and sort of the scale and scope and future plans are.
Yeah, no, that's a great question. So a lot of these studies that we have for stillbirth is very much limited in scope to understand causes or to identify causes. It's a complex phenotype, if you will, a complex obstetric outcome. There are many contributors. And what we've done is just not enough, in research and in the clinical setting. So we want to move from these just preliminary studies to understand the definitive causes. So we want to develop large. So we have a consortia actually formed, a stillbirth consortia, and one of the sites is University of Utah here, among many sites, and supported by the NIH. And so what we want to do is within the consortia, we want to build a quality database that identifies the unexplained stillbirth with a complete clinical workup, and be able to catch up with what other people have done with conditions like type 2 diabetes and obesity and so on, to find predictive biomarkers, what we call biomarkers, early in pregnancy to prevent and guide therapeutic guidelines. And there are some people at the University of San Francisco that have already done with conditions like spinal muscular atrophy, SMA, and other conditions that are caused by monogenic diseases. But we want to assemble all of that information and be able to find a really reliable biomarker to predict and treat pregnancy. That's where we want to go. And to get there, we really need quality data, and we really need to scale up the patient population and data that we have.
And I guess, like, maybe for those of us that are less familiar with the whole process, how much of that will you be able to do with the current NIH grant, and kind of what else is needed beyond that?
Yeah, it's a great question. So far we are on target with we just need about 295 parent offspring families to participate in our study, to be able to. So that's our sample size calculation for our justification of the statistical analysis to find clinically important genes. So far we are sort of on target, but it's not enough, and cost of sequencing have gone down, but the number of people that we have to sequence is quite a bit, and we have to analyze the data, and we're looking for a needle in a haystack. So we're looking at so many genetic variants in the entire genome, and technology is continually advancing, and we're tapping into newer technology to try to identify these genomic regions that we're not able to access with previous technology, and so on. But we're getting there, and it's not always going to be enough when it comes to the funding. But what we're trying to do actually in this project is to try to combine what we have, leverage what we have, and then add additional data from prospective studies.
Or you had a question?
Yeah, I thank you for the presentation. I just was thinking that and it's really for you, Megan, and the doctor, is whether your database registry that RPLA started might provide him with additional people perhaps who would be interested in participating.
Yeah, that's an excellent tee up, Lori. Yes, I think not directly. So short answer is yes. Long answer is sort of is up to the individual. So our registry, which will relaunch early next year, probably February 2026. Anybody that joins that, we have the ability to then, recontact them. And so if there were particular parameters that, you know, Dr. Workalemahu's, if they were looking at a certain profile of patient, we could certainly search our registry and say, okay, this is the relevant population that would, this group of people would be relevant for that study. We could send an email to them within the registry tool and say, hey, encourage you to enroll in this study if you're interested. And so that's the way that they could link.
Okay. Yeah.
And that was actually, I had another question for you as well, because, I know you said, and it is true, right? Like both recurrent pregnancy loss, early losses and late losses, they're all complicated and complex etiologies and causes. But I find it interesting with like the HLA gene in particular, that comes up a lot in kind of when you're reading the blogs or seeing the information about early recurrent losses. And so I am curious to see where there will be an overlap. And, you mentioned UCSF, and I know they're doing some big genomic studies. Like, how do those groups share data with one another so that you can say, hey, they've found this is really relevant to early losses. We've found this might be relevant to later losses. What does that sharing of data look like?
Yeah, it's that's one of the struggles that scientific community is facing right now in terms of sharing the data. In terms of our studies, these are very few families or very few individuals, enough to kind of would be risky to identify. So we wouldn't be able to share the families that we've analyzed in this data. But what we want to do through the Stillbirth Consortium is actually we're at the moment we're talking about collaborating with UCSF and UCSD and trying to, so our objective is actually to look at the placenta, because a lot of the early losses, I haven't shown data from Yale actually that shows a distribution of about 1,400 placentas they looked at. And a lot of the early losses have these placental abnormalities that you can identify looking at the pathology exams, but also the stillbirths after 20 weeks gestation, they have some of them, but many of them they were small for gestational age, meaning a condition called placental insufficiency may have contributed to that. So we're looking at combining data, placental data, with those institutes, and I think the example that I mentioned with I believe there are a few conditions like SMA and other single gene mutations that UCSF is doing, as they partner with companies also to try to identify vectors that they can administer in utero to treat, to look at treatments and things like that. They're advanced in that realm. We are trying to look at the placenta and find biomarkers. Like, for example, people know about cell-free DNA. There is a maternal circulating blood that contains fetal DNA, and that it's called cell-free DNA, and that's actually placental. That DNA 10% placental. So what we want to do is we want to draw blood and maybe test the blood and then find, if the biomarker turns out positive, we want to be able to monitor the pregnancy early, and maybe if there are therapeutic options provided by those institutions, like in UCSF, we want to be able to test for that and provide treatment based on that. I don't think we're far from doing that soon.
We have time for maybe one or two more questions, if anybody has any. Oh, Ben, go ahead. Go ahead, Ben.
Yeah, I'd love so like the work you're doing sounds very promising, very interesting. I'd be curious, with your kind of expertise and knowledge and exposure to work going on in the area of stillbirth and miscarriage, what other areas, maybe beyond what we've talked about tonight, what other areas do you think are most interesting or most promising for future research into causes of or treatments for recurrent pregnancy loss?
Yeah, so we're actually to some of the things, it's in the context of stillbirth in this talk. I mean, genetics in this talk, but we're trying to understand geographic disparities of stillbirth in Utah, looking at what's called a geospatial clustering, and that's kind of one of the things we're doing to try to inform or guide providers, where is the need, and also what factors may be contributing to that. Like, is it access to care? Is it disparity by socioeconomic status? So we're trying to expand that Utah data nationwide, trying to collaborate with other people. And that's been a great experience for us to try to understand the geographic disparities in stillbirths, which has not been really done much, and then overlay that with things like air pollution in proximity to pollutant plants and rivers and things like that. And that kind of helps you map out some of that as well by just doing the geographic clustering analysis. We're also trying to understand the economic burden of stillbirth, the economic and social, psychosocial, which means we're quantifying the cost as well as the intangible consequence of losing a baby, psychosocial impact. So we are fortunate to have a team that helps us model that intangible aspect of it. And so that really helps us to show that, there's a need here that's been overlooked, and stillbirth has not been very much, as also for pregnancy loss, early pregnancy loss, has not been a major funding priority, in the past. And so we want to be able to show there's this burden that also costing families and the healthcare system, and we want to solve it. I mean, in terms of understanding causes, I think there's many contributors to stillbirth, and genetics is part of one of them, but there are many other factors. And the point that I wanted to make in this is hopefully to avoid stigma and blame and things like that. We can't just be looking at maternal, the mother only. We have to look beyond the mother and other contributors. And we're doing a lot of that work. It's just this, it takes all day to talk about it.
Absolutely. Thank you for that. I think agreed with agree wholeheartedly that looking at the whole system and getting away, because it's so easy for, I feel like it's so easy to internalize that guilt and think that it's all on the mom, when that is absolutely not the case. So I appreciate you calling that out and saying it, and I feel that acutely. We've got about 5 minutes left, so I'm going to, at this point, just say thank you so much for your work, your interest in this community, in this space. And as you mentioned, this is not the like hot area for research that all the funding is going to. So I think those special clinicians and scientists who are choosing to work in this area, it's because they really do care about these families and these babies. And so I really want to thank you, Doctor, for your presentation, for your time. It is an honor to be able to help you move this field forward. And so just my gratitude to you. And for everybody else on this call, this organization would not be able to support this work without every single person that is on the call tonight. So that's also my thank you to this community, to everybody that's here listening. I hope that this makes you just as proud as it makes me. And hopefully we'll be able to, it's a 2-year funding period. So last year was the first year that we started to see the results of that first grant cycle. This is coming now year after year. So hopefully we'll be able to do more of these, and you'll be able to see again and again the type of findings that your support is allowing us to help. And
Thank you so much.
I'm happy to stay on the call too, as well. If anyone has questions for me about the organization, that I wanted this to be really focused on the science, but I have nothing after this and can answer many questions about our strategy, our long-term plans, our vision. So I'm always happy to do that at any point.
Thank you so
Also,
much.
thank you.
Yeah, thank you.
Thank you for all the questions as well.
All right. Yeah, thanks, Joey. Thanks, Amy. Thanks, everybody. I know it's late on the East Coast, so if people need to pop off, please do that. I'll stop the recording at this point and just stay on in case anyone wants to say